do you use other tools to study microglia, for example, or these are the primary things that you're doing?
Right now, during the postdoc, over the last 4 years, as a primary readout, we actually use single-cell RNA sequencing mostly.
Another very fancy technology.
Which is basically looking into transcriptomics, but on the single-cell level. So we have a much more focused approach on and see on a single cell level what's happening within the brain or within each microglia in the brain.
Why is this important? So you can look, let's put it things in perspective. So let's imagine our brain, let's say the hippocampus, right, where the memory is formed and etc. Very important brain region, and you have microglia cells there as well, not only neurons and other glia cells, right? When you have now single cell RNA sequencing or sick, what do you mean with single cell? You point one cell and you know in this particular cell what is being translated, transcribed, or you have a bunch of cells that you actually test or examine afterwards?
Yeah, so what happens there is basically, if we stay with this hippocampus example, we would take the hippocampus, we would prepare a single cell solution. So we would basically dissociate the tissue into a single cell solution, which can be done by different methods, and different other friends of ours have also shown which methods to use in order to preserve the transcriptome in the best way. So that would be manual dissociation on ice versus enzymatic digestion, which actually alters the transcriptome quite a bit. So this is not really something that is recommended at the moment in the field. With this single cell solution, you would then go into a single cell RNA sequencing workflow where you prepare actually from each single cell a library that then gets sequenced and then also computationally analyzed. So there are methods right now available where you can look into what you were saying, more spatially correlating transcriptomics with actually the position of each single cell more or less within a given brain region, for example. So these are very advanced methods where, for example, where you do spatial transcriptomics, there are several providers available to do that.
So in a sense, when you say single cells, it means like single cell type, right? Not one cell, but like all the glia that you dissociate from the tissue. Is that correct?
So what we would actually do is we would dissociate the hippocampus, we would isolate microglia. So we have only microglia, and then we do transcriptomics on the single cell level of every microglia. So in the end, we actually zoom into the population of only microglia, and then we can really look into each single cell. Like imagine you zoom into, the brain is Germany, you zoom into one city like Göttingen, and which would be the microglia, and then you zoom further into every person that walks on the streets, and you can assess, so to say, their characteristics, which then would be single person studies. So that's what we would do.